Swayam Prabha. Digital India. Educated India.
I'm Swati Tiwari. I'm a faculty at the School of Biotechnology, Jawaharlal Nehru University, New Delhi. And this project was supported by DTN Swayam Prabha, MHRD, New Delhi. Okay, so far in the last chapters that we have talked about, whenever we are talking about cloning, we are talking about cloning a DNA fragment or a gene into the cloning vectors. Now, in this chapter, we are going to talk about what are gene libraries. So, we'll describe what exactly are gene libraries and what are the kind of questions that researchers ask for which gene libraries are needed. We will also discuss the main types of gene libraries and how to construct them, and some very basic techniques for screening of these gene libraries. So, gene libraries are also known as gene banks, and essentially they are just a collection of DNA fragments from an organism that represents the entire genome of that organism. So, when you make gene libraries, then you have to do a quality check to make sure that it represents almost the entire genome of the organism and you're not missing any part of the genome. And based on the types of starting material that you use, they can be of two different types. One is the genomic library, which basically is prepared from isolating the entire genomic DNA of any organism. And the second type is the cDNA library, where you isolate the RNA from an organism, convert it into complementary DNA or cDNA, and then prepare the library. So let's now discuss in detail about these two types of libraries. But before we do that, we have to understand what are the questions that require one to create a gene library in the first place. So the first use of the gene library that one can think of is to create a physical maps of all the genes that the organism has. Where are they located in the genome, and what is the context in which they are located? So you might have studied in genetics that the distance between the genes is measured in centimorgan units. Now those distances were based on the genetic analysis and focused on whether the genes are being segregated together or independent of each other. Based on that, this genetic analysis, one could create the genetic map and say, okay, these two genes are probably close together and these genes lie far apart in the genome. But to create a more detailed and a more accurate physical map, one needs to start with creating the gene libraries so that you can sequence these fragments and then recreate the entire sequence so that you can study all the elements that are present in the genome. Secondly, if you want to identify genes which are related to a particular cellular function, for example, so you might have studied in cell biology that all the cell cycle genes, CDC genes that were discovered early on in either S. pombe or S. cerevisiae or even Xenopus, they made use of the gene libraries. Similarly, for example, you want to study, for example, how a protein, a secretory protein, is targeted to the endoplasmic reticulum. Obviously, all these functions require participation of many, many genes. So, how do you figure out what all gene products are required for a particular cellular function? And you might have studied about the sec mutants in yeast, where people used the cDNA expression libraries in yeast to complement the defect in certain functions, and then they can figure out which genes are required for a particular cellular function. Similarly, when you are studying a particular disease process, for example, you are studying cancer, and then you want to compare what are the genes which are expressed aberrantly, or whether there's any new gene expression in the cancerous tissues. So you can create the library from the diseased and normal cells, and then you can do a lot of many different types of tricks to understand the differences between the two types of libraries and the sequences. Disease processes also, sometimes you want to make genome-wide association studies, GWAS studies. So for example, you want to compare whether there are certain single nucleotide polymorphisms in individuals which may be making those individuals prone to develop certain diseases, for example, diabetes or obesities. So for that, you need to do genome-wide study of sequences, compare all those sequences in a population and figure out things. So again, gene libraries are very useful in order to do those kind of studies. Of course, you may be aware that nowadays many sequences from many different organisms have been carried out, and they are all publicly available to researchers. And the starting point for all those genomes to be sequenced is the creation of a good quality genomic library first. Similarly, you may want to do a comparative genomic analysis. You may want to compare the genome sequences of, say, early eukaryotic organism like, say, entamoeba, with something which is, really higher up in the eukaryotic ladder, like a human cell. So you can do a lot of comparative genomic analysis, and for that also you need to create gene libraries first. Of course, the drug discovery platforms, they have made tremendous use of gene libraries, and there are many innovative protocols that have come up in the area of drug discovery using the concepts derived from the gene libraries and DNA sequences. Similarly, if you want to test, for example, the specificity of a particular drug, you have made, for example, a drug against a protein X, but you are not sure whether it binds to any other protein in the genome. So you can take an expression library and you can test your drug against all the proteins that are expressed by that particular cell type or that organism. Similarly, the microbiome research. So you know that many environmental niches, they have very complex, ecological milieu, where there are so many different types of organisms. Now we don't know how to culture all of them. So culture techniques for many organisms are still not there. So what people do, they create a metagenomic library. So they take the environmental sample, they extract the entire DNA from those samples, and then they make what is known as a metagenomic library, so that they can later on understand what are the different types of organisms that are there in that sample, what are the, whether they are, what is the gene component, what are the genes that are present, do they have similarity to other things, and so on and so forth. Similarly, our gut flora, is a very complex flora, And so metagenomic libraries and their association to health of an individual is also a very current field of study. So all these very broadly, I have tried to cover different uses of the gene libraries. Of course, there are many different applications, and there are many ways in which the gene libraries have been used and adapted in order to develop more advanced and modern technologies. But right now we will just consider on very basic aspects of gene libraries. So let's first go over the major steps involved in creation of a gene library. And of course, the first step is to isolate the genomic DNA. Now, most of the genomic DNA isolation protocols are pretty standard protocols, but sometimes there are minor technical details that might differ from one organism to the other. But essentially, you have to first isolate genomic DNA, and then you have to fragment that genomic DNA. Now, this can be done by just sharing the DNA, chromosomal DNA, or you can digest the chromosomal DNA by restriction enzymes, so that you have a collection of all these different DNA fragments in a tube. So essentially, the entire genome of the organism is now present in form of these fragments in a single test tube. Now you take a vector which has been, if you use the restriction enzymes for digestion, you can digest the vector DNA with the same restriction enzyme or two enzymes, so that the fragments can be cloned into these vectors. If it has been done by shearing, then you can blunt end the fragments and then clone them. So whichever way you do, you basically then use a DNA ligase to clone all these different fragments into the vector in a single reaction. So you have digested vector in a test tube, you add all the fragments from the genome that you have in that test tube, you use the DNA ligase and let the reaction go. So essentially, in all probability, most of these vectors, molecules will have one fragment each that is cloned into them. So then you take these collection of recombinant vectors and then you transform E. coli cells. Now obviously, because depending on the genome size of an organism and the length of these fragments, there will be a large number of potential E. coli cells which will represent the entire genome of that organism. So let's now look at some of the relevant numbers and think about what are the considerations that you have to deeply think about before you take up the challenging task of construction of a genomic library. So nobody makes a genomic library, very casually. It's not a trivial task. It's a pretty challenging task which requires a lot of technical expertise, and therefore it is best if you understand at least some of the theoretical considerations before you take up this challenging task. Now this table here shows the genomic size of various organisms. I have shown E. coli, yeast, S. cerevisiae, Arabidopsis. This is a model plant that is very often used in plant research, and of course the human genome. And as you can see that E. coli genome is about 4.6 megabase pair. And as you go through these different genomes, the size of this genome has increased. But once you come to the Homo sapiens, it is 3.2 gigabase pair genome. What that means is 3.2 into 10 to the power of 9 base pairs. So that is how big a human genome is. So how do you really tackle such large genome to make the libraries and what are the considerations. So let's first see, supposing you decide to use a restriction enzyme to cut these genomic DNA into fragments. If you use a restriction enzyme whose recognition site is 6 base pairs, then the probable frequency of finding such a site in the genome can be given by 1 by 4 to the power of 6, which is once every 4096 base pairs. And this number we arrive at, this kind of calculation can be done, and which assumes that all the 4 bases have an equal distribution in the genome. Then only you will have all the 6 positions at every base, you have the equal probability of 4 different bases to be present. And therefore, this is a theoretical number that we arrive at, that the likelihood of finding a 6 base pair size is once every about 4 kb fragment of the genome. Now, if you use this number and divide the size of the genome by the average fragment length, which in this case is 4096, then the minimum number of clones which will be representing the entire genome of the organism is given here for each of these organisms. So for E. coli, 1,000 clones will represent this entire genome of the E. coli. So this is the minimum number I'm talking about. This number obviously will increase as the genome size increases. And for human cells with the 3.2 gigabase pair genome, the minimum number of clones that you will require to represent the entire genome will be 750,000 clones. So that's a huge number. However, when we do this theoretical consideration, then we have assumed that at all positions the nucleotides are present in equal, there is an equal probability of all the 4 nucleotides to be present. However, many organisms will not have all the nucleotides present in equal numbers. So there are AT rich genomes, there are GC rich genomes, and so on and so forth. So that leads to not having the theoretical calculation to be observed in practice. Similarly, many restriction enzymes, they are methylation sensitive. So if there is a methylation of C residues in the genome, then many sites, even if they are there, they may not be digested by the restriction enzyme that you have chosen if it is sensitive to methylation. And because of these 2 points here, the actual number of fragments that you generate from a genome is quite different from the predicted number of fragments for any given restriction enzyme. So to give you an example, if you take NOT1 enzyme, which is an 8 base pair cutter, then the predicted number of fragments of E. coli genome are 72. However, when you actually digest the E. coli genome by NOT1, the actual number of fragments are only 20. So there can be discrepancy between the theoretical numbers and what you actually get on the bench. So then, which restriction enzyme should we choose? So one consideration that is very important to remember is that we create genomic libraries so that we can sequence them and arrive at the entire genomic sequence of that organism or the sequence of the chromosomes that are in that organism. So in order to do that, because restriction enzymes will cut the genome in many fragments, you want to have the clones in such a way that there is an overlap between the clones, right? So ideally, if you can get one clone to represent one gene, that is the best case scenario. But we know that eukaryotic genes are very, very big. The restriction enzymes will cut indiscriminately between the gene and in anywhere. So you will at least try to achieve to have a specific genomic region to be represented by at least a few overlapping clones so that you can reconstruct the entire genome. So a 4-base pair cutter enzyme is generally preferred, and one of the commonly used enzyme is SAW3A. And the reason for choosing a 4-base pair cutter is as follows. That we know that the frequency of a 4-base pair cutter site to be found is every 256 bases, assuming the equal distribution of nucleotides. Now, this DNA fragment here shows it's a human DNA fragment, and these are all the SAW3A sites that are present. Now, the scale is given here, 2 kb. Now, you can see within 2 kb there can be multiple sites, but they are not exactly evenly distributed, right? And that is because of the bias, codon bias of the genome, which may be more GC rich. So if we let SAW3A to cut this genomic fragment completely, then there will be many small genomic fragments, and it will be very difficult to reconstruct the sequence of this entire fragment. So a trick that is used by researchers to tackle this problem is to carry out what is known as a partial digestion. And this partial digestion is nothing but you dilute the restriction enzymes quite a bit and then add very little of that enzyme, and then you withdraw the samples and keep checking to obtain the fragment size which are desirable. So for example, if you want to generate about 20 kb fragments, then you have to basically set up the digestion reaction and standardize as to how much SAW3A and how much time is required to digest a particular genome into about 20 kb fragments. You can check this on the agarose gel. So what happens during a partial digestion is that not every site will be digested by SAW3A. So it will be making random cuts at different positions, and because of that random cuts you will obtain overlapping fragments. So in a cartoon it is shown here. So supposing this site is digested, then maybe this site is digested, this site is digested, this site is digested, and rest and this. So you generate all these different fragments, and because they are overlapping with each other, when you sequence all these fragments, by aligning all of them together, you can get a sequence of this human DNA from this end to this end. So this is what researchers try to achieve when they are creating a genome sequence. Now, in reality, the actual number of clones that are needed to be very certain of finding a particular sequence is much higher than the minimum numbers that I talked about before. That, okay, for a human genome, if you have 750,000 clones, then it represents the entire genome. So why the actual number is much higher? The actual number of clones is much higher because, as you saw in the previous slide, the cuts that are made are random. Now, when you are screening the colonies, how do you make sure that you will find, say for example, this particular fragment, right? You can only find it in a random, so you cannot possibly check all the colonies that are present in the genome library one by one. And so you want to spread out your genome library on plates, and then you want to, so maybe you have 10 or 15 such plates, and you want to find the gene of your interest. So because it's a random choice, therefore the actual number of clones need to be much higher to increase the probability of finding a particular gene of interest. So people have done this exercise theoretically, and they have come up with a formula which basically tells you what are the number of clones which are required. This is shown by n. so that you can find a particular sequence with a certain probability, which is P. So n is equal to natural log of 1 minus P, the probability with which you want to find the sequence. So for example, you want to be 90% sure that when you screen the library, you get the clone of your interest, then P becomes 0.9. You want to be 99% sure, P becomes 0.99. So natural log of 1 minus P divided by natural log of 1 minus F. So what is this F? F is nothing but the fractional proportion of the genome, as I calculated before, and this is basically the size of the fragment divided by the genome size, and this number you substitute in the fraction proportional of the genome. So now let's see what are the number of clones that are required and how it changes with the probability with which you want to find the sequence and the fractional proportion of the genome. So in other words, how do you choose the fragment length and what consequence that fragment length has on the number of clones that are required. Okay, so let's take a genome, which is human genome, so 3 into 10 power of 9 base pairs, and we consider two different fragment lengths, 4096 base pair and 20 kb fragments, so 20 into 10 power of 3 base pairs. Let's put P at 99%, so you want to be 99% sure of finding any sequence that you are looking for in the gene library. And so the fractional length will be, in this case, 4096 divided by the genome length, 3 into 10 power of 9. And in this case, the fragment length is 20,000. So F will be 20 into 10 power of 3 divided by genome length 3 into 10 power of 9, which is 0.6666 into 10 power of minus 5. Now you put these values in the formula. N is equal to natural log of 1 minus P divided by natural log of 1 minus F. So when you do that and do the calculation for these two cases, so if you have a short fragment length of about 4 kb, then you need to screen 3.3 into 10 power of 6 colonies of a human genomic DNA library in order to be 99% sure of finding a gene of your interest. As you increase the fragment length to 20 kb, now the number of clones comes down marginally. It becomes 6.9 into 10 power of 5, which is definitely lot less than 10 power of 6, tenfold less. So because of these considerations, vectors that can take larger inserts are preferred so that the number of clones that are required to be screened is much lower. And because of that, instead of using plasmid DNA libraries, people prefer to make genomic DNA libraries into either lambda phages based vector, which can take anywhere between 10 to 40 kb fragments depending on the kind of lambda vector that you are using. And so, for example, if you're making E. coli genomic DNA library, you can probably manage with that. But with increasing genome size, maybe you need better cloning vectors in order to reduce the number of clones that you need to screen. So you can use a P1 bacteriophage based vector, which is P1 artificial chromosome or PAC vector. It can take anywhere between 100 to 150 kb fragments, or you can use bacterial artificial chromosomes, which will take about 300 kb fragments, or yeast artificial chromosomes, we have talked about it before, which can take up to anywhere between 1000 kb or even more fragment that can be cloned into these vectors. So these vectors are preferred to make the genomic DNA libraries, and as the genome size increases, then you want to use a vector that can take much larger fragments. So let me just remind you about the lambda vectors. And you remember there were two types of vectors, lambda insertional vectors and lambda replacement vectors. And while the lambda insertional vectors can take up to 10 kb fragment, the lambda replacement vectors can take up to 40 kb fragments. So essentially both the vectors, when you clone the gene into them, they result in the inactivation of CI gene. And this CI gene, if you remember, was important for lysogeny. So when you disrupt the CI gene due to cloning, then the recombinant vectors will basically undergo the lytic cycle. So when you, after cloning, you infect the E. coli host, and so both non-recombinant and recombinant colonies, bacteriophages will grow. And so the wild type will mostly be lysogenic, whereas recombinants will always be lytic. So you get a cloudy plaque where it is the non-recombinant bacteriophage vector, and the clear plaques because of the recombined bacteriophage vector. And a special strain like HFL strain of E. coli is used, which basically stabilizes the CI gene. So all non-recombinants will not produce a plaque because they are lysogenic, and all the recombinants, because they don't have the CI gene product, they will all produce lytic colonies, and therefore they can generate the progeny. So you will have clear areas on the lawn of E. coli cells, and those plaques, because they have been generated by lysing the E. coli host cells when there are enough number of bacteriophage particles that are produced. So those clear areas or those plaques will contain these bacteriophages which contain the recombinant DNA. So with this background, now let's talk about what are the various ways in which the gene libraries can be screened. So there are three different basic approaches. One is the hybridization analysis, second is the PCR based analysis, and third is the immunological technique, which is used for only cDNA expression libraries and not for the genomic DNA libraries. So let's first talk about the hybridization analysis. So in this case, you have a master plate where you have plated out your recombinant E. coli on the agar plates. In reality, you don't have only a single plate because there are large number of clones that are there that will represent the entire genome. And so if you put them all in one plate, all you will get is a lawn. So you want to spread it out into different large number of plates so that you can get well separated plaques. Now you take a nitrocellulose membrane. So I will just show the cartoon for one single plate, but essentially you will have a collection of plates on which this analysis will be done. Excuse me. So you take a nitrocellulose membrane and you gently put it on top of the plate, and you lift it very gently so that the plaques, they do not get dragged and mixed with each other. And you also want to mark the orientation of the way this nitrocellulose has been put. Normally people just make a couple of punches in 2-3 different places so that they can align it later on with the master plate. So you lift the nitrocellulose, and now what is going to happen is all this plaque material is also transferred onto the nitrocellulose membrane. So you reserve this master plate, you keep that, you store that properly, because then you will need to pick this plaque depending on which one of these are actually the clone, represent the plaque of your interest. So now, because the plaques have been transferred and they will contain the bacteriophage vectors containing the DNA of interest, right? So each of them has a different DNA fragment that is there. So you lyse them, denature the DNA, and hybridize to a DNA probe. And if this DNA probe is labeled with radioactivity, you do autoradiography. Nowadays you can also use a fluorescently labeled probe, and you develop it on a film, photographic film, or phosphorimager. Then the probe will hybridize to whichever plaque contains the gene of interest. So supposing one plaque lights up on your autoradiograph, so this will be one signal, and most likely this is the one. So now you align this nitrocellulose with the original master plate, and you know this is the plaque that contains the signal. So you go ahead and you pick this plaque from the master plate, which will contain the bacteriophage with the desired DNA fragment. You isolate the DNA and then you identify the gene by sequencing. Now, similar kind of analysis can also be done for bacterial colonies, and that is done by what is known as replica plating, where you have a velvet covered head. It's like a rod that you gently press on the master plate and then plate it on several different plates and grow them in order to have a replica of all the colonies that are present on the plate. And also you make a replica on the nitrocellulose. Now instead of plaques, these will represent the E. coli colonies. And so you lyse the bacteria, denature, hybridize to a probe in a similar way that you've done before. And instead of picking a plaque, now you align and pick up the E. coli colony from one of the replica plates that you've created from the master plate. Isolate the plasmid DNA and sequence. Now, if you're doing plasmid cloning, then there will be a large number of E. coli colonies. And so the replica plating does not give a very clean results, because the E. coli colonies, they give a background when you develop the film. So picking up what is positive, what is not positive is a little difficult. Secondly, even though you may lift very carefully the colonies, there is a lot of smudging and mixing of the colonies, because E. coli colonies are very, very small. But that can be done if you have very few colonies and they are very well separated on your master plate. Then replica plating can work very well. So essentially what you have to understand is that you can hybridize whatever, whether it's a plaque, whether it is E. coli containing a plasmid, or a yak or a BAC containing the DNA fragment of your interest, to a DNA probe. As long as you can do that, you can pick up the original clone or original plaque that contain the gene of your interest. So these probes, they are of two types, the homologous probes or heterologous probes. Homologous probes, as the name suggests, they are from the same organism, and these are the DNA fragments which are the exact complement of the sequence that is being searched. So for example, you have a partial fragment of a gene. Say you have a mouse gene, you have a partial fragment of that gene, and now you want to pull out the entire gene. So you make a probe out of that fragment that you have and screen a genomic DNA library, and you can pull out the entire gene. This is also done to identify the genomic location of different genes. Also, if you have the genomic library from normal and diseased tissue, then you can use the probe to pull out those genes from the normal and diseased tissue, sequence them to study what type of mutations might be there in the disease condition. The heterologous probe is similar to the gene being sequenced, to being searched, but not totally identical. And this is generally done to search for homologs of genes in different species. So for example, you may have a gene sequence, a gene fragment from a yeast gene, and you want to pull out the homolog of that gene from, say, a mouse cell. So you prepare the probe of the yeast gene fragment, and then you search a mouse genomic DNA library and pull out the gene which is homologous to the yeast gene in this manner. So these are the different kinds of probes that can be used, and of course the probe has to be labeled so that it gives a signal and you can pick those signals. One thing I forgot to mention in the last slide, in general, it is not that you get only a single positive plaque or single positive colony. In reality, you get multiple colonies or multiple plaques, because you remember that the genome has been fragmented and there are many overlapping fragments. So you may get several of them, and then you may need to repeat with a slightly different probe in case you feel that some part of the genome is missing. Okay, so these probes, they are usually labeled, so one can use 32p labeled probes or fluorescent probes. Nowadays there are companies which can make the fluorescent probes for you at a cost, but whichever way they are labeled, you can use a number of techniques, a number of tricks to label them. You can either use nick translation, so that the probe is labeled at multiple places, most likely. You can also do an end labeling of the probe, so that, for example, 5' end of the probe contains the label. You can also use random priming and PCR method in order to generate a probe which is labeled. The other method of screening is the PCR based screening, and this is much more labor intensive because it works on the principle of dilution method for colony identification. So here all the colonies, they need to be transferred to these 96 well or 384 well plates. Of course it can be automated, so one can automate these steps also. So in essence, what you do, that after transformation of the E. coli, the colonies are picked and you can pool certain colonies in each of these wells. So for example, you may decide to pool 50 colonies per well, and then you plate in a 96 well plate. Then you isolate the DNA from all these wells, and then you do a PCR using gene-specific primers. Now, because of the overlapping nature and because the same fragment may be present in different overlapping clones, you may get a number of different clones which light up upon, which basically are positive in your PCR reaction. So you pick these positive wells, and because initially you had 50 colonies in each of these wells, now you dilute each of these. So supposing there were 50 colonies, and if you dilute it, many times in such a way that now when you plate them again, it is 1 colony per well. And then again you do the PCR, okay? You may again hit certain number of colonies which are positive. And so you can repeat this kind of test, dilution PCR several times till you are very sure that you have the PCR coming up from a single colonies in each of these wells. Then you sequence them and recreate the entire genomic sequence of that region. So this PCR-based screening can also be automated and done much more quickly. So to summarize, this part of this lecture is that I talked about the gene libraries, which is nothing but a collection of DNA fragments which represent the entire genome of an organism. And these fragments of the genomic DNA can be produced either by shearing of the DNA, say by brief sonication, or partial digestion by a restriction enzyme. And in general, a 4 base pair cutter is used so that many overlapping fragments are generated. And cloning a vector could be either a plasmid, which is not preferred because you generate too many clones that need to be sequenced and screened. But for larger genomes, bacteriophage lambda-based vectors or BAC vectors, PAC vectors, or YAC vectors are used. And of course, the choice of the cloning vector will depend on the genome size. The screening of the genomic DNA libraries can be done either by DNA or RNA hybridization or by PCR-based methods. And genomic DNA libraries are very, very useful for genome mapping and sequencing of the genomes, for finding novel genes. For example, sometimes, in many cancers, the genes which are not normally expressed can be expressed. Another complication with cancer is that not all genes are expressed in all the tissues. So when you're dealing with a particular type of cancer, then the library prepared from that particular tissue is more informative compared to a library prepared from any other random tissue. So genomic libraries, they are important for finding novel genes and also for finding homologous genes, and also they are very useful for finding the drug targets. So for this part of the lecture, you can refer to these two books, Molecular Cell Biology by Lodish et al. Or Molecular Biology of the Cell by Bruce Alberts. Both these books are available on the NCBI website at NIH. You can Google them and find them. Their full text is available online and you should be able to study them. So please note down these two resources. And you can search for these resources on I think it is org. I'm not too sure about this, but you can search for NCBI, and from that link you can find these books. So in the next lecture, then I'll be talking about the cDNA libraries.